Welcome, everybody, to Learn With Lowell. Today, we're joined with Collin Ewald, assistant professor at the ETH of Zurich and founder of the Swiss Society of Aging Research. We have a number of really great things we're going to get into today. It's going to be about longevity, health span, obviously Collin's work. Collin, welcome to the show.
Thank you so much. Thanks for having me.
Yeah, and I believe you're in Switzerland, which makes sense.
Yes, I'm based in Zurich. It's all the stereotypes. It's beautiful here. I'm originally from Switzerland. I was born in Switzerland, and when I was younger, I thought it's super boring. So that's why I did my PhD in New York. Much more exciting there. And then I went on to Boston, and now I'm back in Zurich. So I miss the big cities, but yeah.
Is Zurich the one with Einstein's clock that helped him with relativity, or is that a different Zurich?
You asked me something there.
If someone knows, I was reading Walter Isaacson's book on Einstein, and I think Zurich is where there's like a clock, and he thought of relative. There's a clock and then a train that helped him spark that. But
exactly. But I don't remember, was it Bern or was it Zurich? So that's why it's like But
I feel like it was sure. Yeah, anyone who knows this definitively, please let us know in the comments. I was just starting with your research. Can you define for us what an extracellular matrix is, just for people listening in? I've been told that sometimes I just dive in and people are like, well, Lowell, can you just define these terms? And then we can learn about them. So what is an extracellular matrix? And then we can discuss how it relates to your work.
Perfect. So, cell and organs, they are surrounded by proteins. And those proteins form like a meshwork that hold everything together. And that's the extracellular matrix. So each cell produces its own extracellular matrix, each organ, right? So it makes the proteins, they're secreted, they're outside, and those proteins come together, like some form like a fishnet and then form around it. And it's actually, there's a lot of things going on. It's pretty crowded. You can think of an information highway. So there are, hormones going through or signal going through, but also it's important for the stability of the cells and also for the physical properties. So shock is absorbed there, right? And it holds things together. And that's why it's also called the connective tissue.
Right. And then we're talking about pretty microscopic things. How do you measure them when you're doing your research? Like what type of weights and measures do you have to account for just to do the research that you're doing on the animal that we're going to talk about in a minute?
Yeah, so if you want to look at the composition of the matrix, so which proteins are in there, we use a technique called proteomics, which tells you just what kind of proteins there are. And if you want to look at the stiffness of the matrix itself, you can do this with electron microscope. You can pinch down and see like how much intention you can get. We also use reporters that are proteins that have some fluorophores tagged with it. So we can see them with the microscope, and then we can also tell how far they're apart, right? So once we stretch a tissue and organ, we can see those reporters being close together or further apart, right? So that gives an idea of the dynamics. And so that's how we look at the matrix itself.
This is like a meta question. I'm always curious with research. Do we have the tools to research it as fully as you'd want? There's an element of like, the Einsteinian imagination when you're thinking about how these systems can work together. And then you're limited by the tools to some extent, I feel, and doing research sometimes. But when you imagine what you want to be researching or what you want to find out, do we have the tools to allow you to delve as deeply as you want to achieve your outcomes? Or do we still need to develop tools to make it easier for you to do your research? More effective. Yeah.
Of course, you don't have all the tools you want, right? Yeah. So usually there's one saying from Sidney Brenner, which I really like, right? Which basically, progress in science depends on new methods, right? That gives you new ideas, the new concepts, and then make a new discovery. And he said probably in that order. So basically, the better the methods, the technique you have, the better you can, come up with new ideas, because nature is so exciting. I tell you one problem we are facing at the moment. We also look at the cross-linking in the matrix between collagens, which is very important during aging or diseases like diabetes. And I wish we could see those cross-links, right, directly. That would make our life much easier. It's always indirect, right? And so if we would have the direct detection system for that one, it would be perfect, actually.
Are there groups or research projects out there looking to give you that fidelity? Is there anyone out there that you know that is working on giving you that ability, or is that is there anyone alive working on that to give you that?
Yeah. No, there's a lab in Yale that tries to make antibodies to specific parts of these cross-linking things. So with antibodies, you could it's one way of detecting it. And then the other people that work more on the mechanical sides and trying to figure out, in microscopy and other things to really see those changes. So yeah, there are efforts being done, but I think only a few. There could be more, I would say.
Yeah. And if anyone, I'm going to keep saying this, but if anyone has any, you never know what people know. That's the cool thing. There might be someone out there who's like, oh, we've been working on this in our lab, at UW-Madison or something, and we just don't talk about it. But for the extracellular matrix, how does it relate to aging? So how do we make the connection between the matrix, aging, and health span?
Yeah, that's the big question we also addressed and tried to look at. So I would start. I'm going to start the way I stumbled into it, because I was not working on the extracellular matrix. So I was looking at interventions that increase the lifespan in a tiny model organism called C. elegans. And there, what came out in gene expression profiling was actually that they upregulate collagen extracellular matrix proteins. And at that time, it didn't make any sense. Why would this happen at all, right? And it took quite some research to stumble in there. So basically what I found is that these upregulations or these things for the extracellular matrix are required for longevity and also sufficient. And so if something is required and sufficient, that means there must be a mechanism, right? And then the next question that came is why would this intervention actually start to repair the extracellular matrix? And for that, I have to show you what's going wrong during aging. And so I'm trying to do this. I'm going to share my screen real quick because it's easier for me to visualize it a little bit.
And everyone listening in, let us know if you like this. We're trying something new today. We're actually going to look at a paper while discussing it. So for visual people and for people listening, we'll describe it and stuff. But just let us know if you like this experiment, because we're both flying by the seat of our pants on this one. But it looks good so far.
Okay. Do you see my screen? Yeah.
Yes.
Okay. So I tried to conceptualize this with this figure here. So that's one of the recent papers. Oops. That didn't help much. That didn't help at all. So
we can see it
from the journal. Okay, yeah. So the idea is, as I mentioned, every cell makes their extracellular matrix. So this should depict a cell. So what you see is a cell. You see a cell nucleus, and the cell transcribes the collagen gene. It's synthesized. It's secreted. It's laid in a matrix, right? And it's integrated. So it forms the matrix, and that's the function of a collagen itself. Now, if there's something goes wrong in the matrix, then what needs to be done is actually that this part is like a fishnet you have, and there's some chunk in there. So you need to cut around the fishnet, right, and get this out and degrade it. And so there are enzymes called MMP, so metalloproteinase. They do this, and then it's internalized. And then it starts again to synthesize new ones to repair it. And that's a process that works very well when you're young. But of course, like many processes, that declines during aging, right? So I give you an example. If you go into the gym and you train your biceps, your muscle will grow, but also your extracellular matrix has to adapt to it, right? And so there's always an interaction between what is your cellular need, what do your cells need to adapt with the extracellular matrix. If you go running, for example, your Achilles tendon will adapt to the running and remodel the extracellular matrix. Now, what we found is that this process declines during aging. And so what we've seen is these longevity interventions, that they just keep this homeostasis on for longer. And the reason is that, during aging, what's going to happen is that, trying to make this a little bigger, but
I can see it pretty good.
Okay. okay, what's happening during aging is that these blue lines here, these should be collagens. They become fragmented with aging. So these are like metalloproteinases that go wild, right? So, sometimes these things are deregulating aging. So they go wild. They're like little Pac-Mans. They start to snip up the matrix into the pieces. So that's called fragmentation. And at the same time, what's happening are these sugars that attach to collagen. So if you have extra sugar in your blood, right, it's somehow it likes to attach to these collagens, right, and they are called these H-glycation end products. And these H-glycation end products, they like to cross-link. So you get this cross-link of collagens. So basically, what you get during aging is a stiffer extra matrix. So it's closer together, but it's also mechanically weaker because certain parts of the matrix are chopped off, certain parts are just cross-linked, right? So you can see the function of this surrounding is just impaired. What's also happening that there are proteins that start to aggregate in the matrix, like chunk, right? Starts to aggregate, and we are unsure whether that's a mechanism by the cell to safe store it or it's actually to remove it. And so, and that's basically the parts our lab has been working on and trying to tackle. And we think, and that's a hypothesis, we still are trying to show that you can fix all these problems if you maintain the exomatrix homeostasis, right? This circle, right? Usually the circle either it goes wrong, there's too much MMP, you get too much fragmenting, or there's no remodeling happening anymore, and then have these H-dependent sugars are added that start to cross-link. And so yeah, that's the what I wanted to say in a nutshell. And so that's why it was easier just to show you the graphic to get some visual representation.
No, it was actually quite nice. I like that. I'm going to definitely see about adding that in the future because it is easier to visualize things. At least for me, some people are they have different reasoning skills. But to what extent is this process? Is sometimes when people as they age, they kind of get set in their ways and they're not exercising as much. How much is that component versus like the chemical changes that you can't rebound from? Because I hear that when people exercise as they age, they can get some strength back. But if we're talking cellular, maybe it's not something that really changes.
Well, let me see. This is a chicken and egg question, right? What's the cause and consequence? So let me say this way. So one part of the aging of the matrix. So there are certain collagens that are made once, secreted, and then if there are no cells around it, they stay there forever. So the half-life time is 114 years, for example, right? And those ones can accumulate all those damage, especially these sugar additives, right? These glycation end products. And then that part you could think of is like if you have a plastic or rubber, like a bucket or a rubber band, right? Let's take a rubber band, right? So if there's too much sunlight and there's some chemical aging with it, it gets brittle with time, and then it cannot hold the forces anymore. And that's one part that's definitely happening, right? And basically, it's unclear how to repair that because the cell is not doing the job. For that, you would need these cross-link breakers or something out there that could remove that in a sense or make this more useful if you cannot replace it. The other part is with the cell, right? The cell can replace the extracellular matrix. And so if it's a tissue there are cells around, it's technically possible. But then again, during aging, those cells not become lazy, but they don't function as good as anymore. Some of them become senescent cells, right? They secrete lots of factors and immune response, and things go wrong there again. And that also leads to changes in the extracellular matrix itself, right? However, if you could rejuvenate the cells, you would also rejuvenate the extracellular matrix. And we see this indirectly, so not our group, but other groups. When I look at these papers done for the rejuvenation with the Yamanaka factors. So these are 4 factors you can give the cell, and it reprograms. The epigenetic resets their age, right? It makes them younger. And one thing that's happening is actually what you see on the transcription profile. You see this collagen is expressed. So what I would assume, and that's just again speculation or hypothesis, that with rejuvenating a cell, you would also rejuvenate the external matrix. You would kickstart that program again to make a younger external matrix.
Is it your intention to how do you intend to do the rejuvenation? Like, what's the process you're going to go down?
Yeah. So the process we go down is actually a couple of really cool observations. If you take a tissue and you decellularize it, so meaning that you take, get rid of all the cells that are in there, you still have the framework, the extracellular matrix.
Like cellular agriculture.
Yes. And so if you now if you do this with an old extracellular matrix and you put young cells in there, think about like, an ant farm you can buy in the store. The ant farm is just like, it's the scaffold you buy, right? You put the ants in there and they start to, fill this whole thing up, right? So you can think about it. So again, you have the scaffold and then you put an old one and you put the young cells in there. They're trying to refill this and remold this, but those cells stay also then old. However, when you do the converse experiment, which is much more interesting, you take a young external matrix and you take cells that stop proliferating. So they become senescent, right? So you technically say like, they're close to death or they're not useful anymore. You put those senescent cells that do not proliferate anymore into a young external matrix. Somehow the young external matrix reprograms them again and they behave like young cells. So the interesting part is you can do this with stem cells. So stem cells that lost their rejuvenation, their stem cell potential, so to regenerate the tissue, you put old ones in there, you can reprogram them. And this also works with even for muscle fibers. There have been really cool experiments done with rats where they took muscles from an old rat, right, and put it into a young one, and these old muscles had the same strength as the young muscles in the other rat, right? And you can do the converse experiment, right? So you have this rejuvenation effects or the other way around, the reprogramming effect. And the last cool evidence is also that you can take cancer cells and put them into an embryonic extracellular matrix. And the embryonic extracellular matrix reprograms the cancer cells to lose their tumorigenicity, which is astonishing, right? And so what we are trying to tackle or think of is like, what is the signal that comes from this young extracellular matrix, right? And so that's the question we are trying to address at the moment to unlock. What is actually what is the permissive signal to say like, hey, you should reprogram, because we can reprogram cell, right? I mean, this is again with Yamanaka factors, right? But there's some signal coming from the external matrix, and so we're trying to decipher what is that signal.
When you discover the signal, will you use something like the Yamanaka factors, or in discovering the signal, you'll see what builds the signal and then try an intervention around that system?
Yeah, it would be cool to do a double therapy, right? Because with the Yamanaka factor, the one problem there is that you program the cell to into a stem cell like kind of thing, right? So you lose cell identity, and that's one of the biggest problems, right? So you don't want to go too far because every cell has a function, right? So a skin cell should stay a skin cell, not become stem cells or anything else. So technically in combination, what you would have, because the extracellular matrix can determine cell type, right? And so the combination would be actually the ideal thing. I think one or the other is not sufficient. I think you need both.
Yeah, it's interesting the idea that maybe the matrix or the foundation is dictating like the form and function of what comes later. I think there's a lot of I don't know what they used to call these, but there was an episode of Silicon Valley where they called them like blood boys, where like the young people would give their blood to older gentlemen. I think it was meant as more of a joke in the show, but we've seen systems like that. But it's interesting if the structure itself has something there that can influence these types of things versus just like some higher order thing like blood or whatever.
Yeah, so with the plot, that's wonderful work also done in the field, and I just think about the work of Tony V. Scory with trying to figure out what are these factors in the young plot that rejuvenates the brain,
Yeah.
right? And one of the factors are actually called TIMP, tissue inhibitors of metalloproteinases. And again, they act on the extra matrix, and that's one of the factors that rejuvenates the brain, which is interesting. But you made a really good point there. What is in the matrix that would dictate cell identity? And so that other people have done wonderful work there, where you could say you take a adipogenitor precursor cell. So this is a cell that could make fat or muscle tissue, right? And you put them on different surfaces, on very soft ones or very hard surfaces, just putting on different type of surfaces, right? That's the only difference. And depending on the surface they start becoming either fat cell or a muscle cell. And that's interesting, right? It really it says, the physical entities of the environment dictates what the cell will become in cell differentiation. And it also makes sense because different tissues in us have different stiffness, right? Bones are much stiffer than, other tissues, right? For example.
What do you, we recently had Michael Levin on, and he was talking about bioelectricity and some really interesting stuff that he's discovered, and how he can take some cells that were of an eye and put it in the back of a frog's, like a back of a frog, and then the spine actually connects to the eye and develops an eye. It co-ops tissues around it to start developing eyes and stuff. What do you think of that type of work in terms of yeah?
It's beautiful. I mean, I'm a neuroscientist by training, so I'm very, neurons use electrical signals, right? And I have my theory. Like every scientist, they have their own hypothesis and theory. And so what I think is going on, I never actually talked to him, and I should do this because so when he does this, he sees that actually these channels open up and different ions come in, right? I think calcium signaling has changed there. That's one of the things. And when we look at, in our research, when we look at the exomatrix and look at the different stiffness and the different compositions, we look at the downstream signaling, like how does the cell sense this, right? And it comes to a similar signaling cascade there. And I want to talk to him and ask him, different factors we found, whether those ones are also important for determining these kind of things. For example, we've done work there to figure out that actually the mechanical forces changes the gene expression in the nucleus, right? So outside of cell, it changes the gene expression. And there's a factor called YAP1 from the hippo signaling that's really important for that one. And I could think, and also that's, sometimes, there are some other factors that are depending on these ion channels, right? And so it's just so much coincidence that I think there might be something that, what he looks at the electrical signaling, right? He can just spots it around from a different level, right? Instead of the cell sensing it, he just hits the switch already, right? It's like, hitting the light switch directly instead of having something that starts then to hit the switch.
Does the afterwards, let me know, and if you don't, I can see if I can work on an intro between the two of you. But because science, we need to support each other. But I'd love to help that conversation happen, because it does sound kind of weird if everywhere you go, it's I think there's a I don't know if that this is the term that's coming to mind, but it's called transpermia, where in a Star Trek episode there was just humanoids everywhere, and they started realizing that there was an ancient civilization that dropped the DNA that built the building blocks that allowed humanoid figures to exist. So if you see humans everywhere, it's kind of it's a weird thing. If you go out in the universe, you see, well, there's a human. Granted, I think it's actual reason is the prop department only has so much, and we're humans that are doing the acting, but that is weird. If everyone's seen these really weird phenomena, it's definitely something to explore. Does Michael's work influence how you look at what you're trying to do? If he's a light switch and you're at the cellular level, does the light switch influence your understanding of the signaling, which would then give you a better understanding of the matrix?
I mean, in the end, I think everything is connected in a sense, right? And it's pretty difficult. I answer that question in a different way. The way I think about this, what we do in science. So there's a really classical pictures of 7 blind monks, and they're investigating an elephant, right? And so they investigate the elephant from all different sides, right? One is touching the trunk and describes it, right? One is touching the foot, one is touching, the tooth, one is touching another thing, and everybody describes the same thing differently, right? And so I think that's what's going on. I mean, the real challenge is really to take all this and then come up with, the elephant itself, right? And that would be the overall goal. And I think biology is getting there, but again, there's the theory of biology is just coming, right? I mean, we're just way behind physics or other disciplines, right? And it'd be really cool to either mathematically describe this, right? To put this all together, because that would be one language that could be universal. But we're not there yet. And at the moment, these are just loose associations, right? And wild connections. And that would be super interesting to show. I mean, you have to go step by step to map this out. Like, the toothed elephant is the same thing with, the trunk, right? It's just, I don't know how to solve that problem, but that would be cool. I think we should do more on, I think you should do more on theoretical biology. I think so. I like it in a sense because it's so wild to think about it, right?
What's an example of theoretical biology that you think about? I don't get into. I don't know anything about it. So you like, this is a window into your heart, and so that I can go and research it more.
So I work a lot with systems biology in a sense, right? Trying to connect different things, and I think the work that I like the best, because I try to think it from the other side, is thinking about aging itself, and it's a multifaceted problem in a sense, right? And you can start coming up with these relationships between age and you have age here and frailty here, how frail you are. So you start getting these relationships in together, and from there we can start building theories how things age in a sense. It's a little bit complicated, and you see me like very loosely defining because it's haven't crafted in a way I was hoping it. But one thing that it's really cool to look at, it's Uri Alan's work in Israel. There's a wonderful video about it, and he connects this so beautifully from the systems biology, the theory of aging, to the drug discovery, right? With these theories, you quantify a lot of things, you measure it, you build a model, and he was actually able to do the next step to actually predict for a drug target, right? So to change things, for example, in fibrosis, which is something that's bad with the extracellular matrix or the collagens and happening, and he was predicting them just targets, like an autocrine loop there to target with pharmacology. And so it's just beautiful. So the whole thing started by putting cells into dishes and measuring different ratios of cells. So you have fibroblasts, skin cells, and immune cells. You put them together in different ratios, you get different outcomes, but you can also quantify this and start building models, right? And from there, he just took the next step, going into the molecular and into the signal transduction and connected this all this way. So it's beautiful work. So I recommend everybody check it out.
I'm definitely going to do that later today. What do you think? A lot of these systems, when we're talking about learning and making hypotheses, I'm really struck by what's machine learning, AGI potential, and like these ChatGPT type things, these large language models. Do you are you interested in them, or do you see them playing into your work already? Like, what do you think about them?
I think it's playing to everybody's work, right? No matter where you look at.
Yeah.
And
There's like AlphaFold with proteins, right? Like they're everywhere.
exactly. And so we used AlphaFold in the lab as well, because some of these extra matrix proteins, it's very hard to figure out how they look like. And if you see the structure, right, from the geometry, you can geometry dictates functions. So you could think about the function, you see it there. So use AlphaFold. We use machine learning for drug discovery, actually. And so we try to train the machine to figure out how does a molecule look like that induces longevity, right? And then we come up. So we've done this just recently, and we found the mTOR inhibitor, which mTOR is a kinase that's very important for in growth and in aging. So we designed with machine learning a new mTOR inhibitor, and went to the classical route to show actually that we synthesized the molecule. We went on and looked in cell culture, inhibit cancer growth, and then we looked in C. elegans, these little round worms. When you feed it to them, they were able to increase the lifespan. And also what we've done there is you can do also the modeling with the machines, right? So like where's the dogging, where is that happening to the protein? It's really fascinating. To me, it's really fascinating in a sense, and I see this as a new way to screen in a sense, because I've done a lot of genetic screens, of pharmacological screens, and for me, this is just a screening system in the computer. And with every screening system, then you have the next step, you have to somehow validate it, right? And to us, since we're interested in aging and longevity, we want to have an in vivo system to feed it. So we synthesize this, feed it to C. elegans, because the C. elegans, first of all, C. elegans eats bacteria, right? So you put the drug on there, so you have a bacteria-drug interaction. So you already, bacteria can metabolize the drugs, right? So you have that part. And then the whole thing is eaten up by C. elegans that, it has to go to the intestine, and from the intestine has to go to the target tissue where you want to do. And so it has a long journey, right? So there you can see immediately, is it toxic? Does it have any side effects? And then we look at aging and lifespan. And lifespan is such a complicated phenotype if you think about it, right? To slow aging. So if that all works, then we know, okay, we predicted something useful with the machine that actually has a chance to work in vivo. And so the short answer is, I think these machine learning things are super great. They're new tools. You have to learn them, because if you're not, you're going to be overrun. But you also have to find a way to validate whatever the machine tells you what predicts in any way, right? So you have to have the ground truth to say, yeah, this is something that's real or not.
Yeah. I think often when people watch I don't know if you're a fan of Marvel or Iron Man movies, people say that Iron Man's thing is like, oh, the suit or the little, like the power of the sun in my hand or whatever. But it's like Jarvis is like coolest thing, because it synthesizes a lot of his ideas, or like tests them out. And you'd have to have a huge like Manhattan Project worth of team to do what he does by himself in a lab. And I think that's something that I generally think his greatest invention is the Jarvis type thing that helps for everything else. And I think that's something that machine learning has the capacity. Like you had an idea, you had machine learning, run through this process that normally I can't imagine how long it would normally take you to do what you just did. It might take years to find the right one and synthesize it and know it's going to work out. That's like, and now you've done that in a matter of a fraction of the time, and you can develop hypotheses and move forward and have a better understanding of biology from it. Like that's crazy.
It is really crazy. Think about it. I mean, these are years of work crunched into, I don't know, minutes or seconds, right? I mean, it's really like it's so amazing once you have the system. And if you have the AI trained, it's amazing, right? I mean, think about all these wet lab screens and all the money, and if you can do it in silico, so to speak, it saves you. I mean, I would say for that paper we put out, the whole discovery part we've done in weeks, which is just like, and that includes setting up the machine, the coding, right, the training and stuff like that. But now we just can go and say, like, this is what we want, and then the machine will compute. It will get hot. So I have to say, we had to at ETH Zurich is a very technical university. It's like the MIT of Europe, right? It's it's very good for these kind of things, but we were not allowed to crash their servers anymore. So we had to build our own little supercomputer to run this. So sometimes the room gets hot when it's really calculating things. So yeah, but it's an amazing tool, I would say.
Is it what's the logic to build on-prem, your own servers versus like grabbing AWS, compute to do the work? Is it what? Yeah.
So with ETH would have been fine, right? Again, you have to wait to Kion. Yeah, you can buy those things. It's all fine. It's just we were just much more flexible to change things on the fly, and it's just much easier, right? To do. Also, we had a lot of data information to put on there. In the end, what's also important is, if you put it up somewhere onto the cloud, how do you know how good your data is protected, right? In a sense.
Yeah.
So
that's not very much.
No, that's the other thing, right? So I think it's worth to have a trusted system, so to speak.
Mm-hmm. When you build, I'm really interested in this type of stuff, so I feel like we're gonna go just explore this for a minute. But do you do the coding? Do you do the building of the networking layer, or are you like the brain that brings in other people to have a multidisciplinary team that builds it?
I'm useless, right? So I'm just bringing the people. I'm more like a biologist, right? I have an idea, I have a vision, and then it's just there's so much talent around, and so here we collaborate with one of the best machine learning guy. He's in Paris, and it's a statistician, a mathematician, right? And that guy is incredible. So he was doing a lot of the coding, and yeah, it's beautiful. If you have the right people, it's beautiful how things come together, right? And then to me, I don't understand nothing about the coding, right? I mean, I tried once to analyze an RNA sequencing, right, with R, and there was all fatal error and stuff like that, and it's just like at the end, I was like, okay, I better not do this. I just break everything. So I really have no understanding. But the good thing is, if you can talk to different people and understand a little bit what the language is. I mean, you need so much expert knowledge to do something really good, right? You have to go so deep. Nobody can do everything, in that kind of sense.
Yeah, the Steve Wozniak, for people who know, helped build. I'd say he built Apple, like Apple II, Apple Ones. Like the personal computers really came from him. He said that at one point in time you could know the whole system, but now you can't. Things are so complicated that you'd spent a lifetime just to understand one iteration of a product, and you can't do it anymore. So there's just layers of abstraction for every product that exists, which is probably where backdoors and stuff get into products so easily. Like there's just like a person who didn't know any better and just like had a design system or something that was done wrong.
Yeah, and it gets so complex. I mean, yesterday I've been sitting in this workshop. I'm going to digress now here. It was about information, right? So, personal data. So if you walk around, if you just take, Google Tracker, right, to do this timeline, Google Maps, right? You can see where you've been, right? And then you can see where the information is. And so it's just astonishing to me how much information is actually shared with all different things. So if you can go to multiple websites like in newspapers, right, and you look at who is actually getting all that data and get this data sold to, that universe is so complex. I was struck yesterday just seeing all this, right? All these different third parties or brokers or anything. It's just like, I mean, you can scroll down for minutes just to see, like, just you accessing website. What's all happening in the background? So that's a system that now got super complicated. And to me, personally, it would be important to figure out, but what is happening actually with your information? What are you doing? I mean, what you look at, for example. It's just, again, totally digress, but I think that's another an important topic to look at.
No, I agree. It's kind of like the Wild West right now. It's also really complicated. So people who are making laws are really hard to understand it. You know, if anyone's watched the Zuckerberg trial where they were like asking him like, what, Zuckerberg's not, I think, the most human of people. Like he seems kind of like a robot sometimes. But like the lawmakers, I think, are a little bit behind the ball as well, which is kind of sad. But for building a supercomputer, and actually just on the idea of, stop it, my phone's going off. But the idea of like Twitter and algorithms, like you can train it to bring stuff to you that you like, which is nice. But I've also heard that like apps like TikTok apparently just like they're like grabbing everything to get a sense of what you like and then feed it to you. So they're looking at your Google Maps, they're looking at what you do, where you go, which, sometimes I'll say like pizza. Oh, wow, I want a pizza. And then like, guess what? All I'm seeing in ads from that on. That means someone's listening actively, some system. And I hate that. Like, that's so disgusting. So hopefully we go less dystopian, because that's kind of dystopian in terms of like no privacy.
And so the reason why I was doing this workshop is, I'm interested, I'm in the Institute for Translational Medicine. So I'm really patient confidentiality, what's happening with electronic records, right? Also, we setting up different things, like how would be information stored, how it works, right? And it's just shocking that, it's so hard to secure these kind of things, right? And then it's just what's happening all with the data. And one thing in Switzerland, what we're trying to do is actually that the patient, they can always request their patient records and data, right? And then how you get that, how can you collect that, how can you have that electronically, which would be extremely important for the next step in the aging and longevity field, is because, you want to use the machine learning, right? To look at your electronic records and, help with diagnosis of things, or have on-time tracking, maybe with Apple Watch or something like that, right? And that tells you, you should drink some water now, or it helps you like on a personal side. And that's the whole goal, and that would be also the power, right? Using artificial intelligence for medicine, and especially for longevity medicine, right? To stay healthy, to look at your blood pressure, or whatever monitoring you do, right? But it's so difficult to integrate all these information. That's why we're looking at to integrate all this information, because you could use Google Maps, see like how much you walk, or those steps, right? And you want to, as a person, you want to integrate this for your health benefits, right? In the end, and also the idea is the incentive to also get interest in them, because you could stay much more healthier, right? With these kind of things, but it's so difficult, and it's already such a universe of things you don't understand. So you map this one, it's going to be extremely hard.
Yeah. In the US, I was talking to a CEO of an insurance company, which will remain nameless, who was bought out by another insurance company, which is a giant one, that the data from the first company on customers, the parent company the new parent company that bought them couldn't access the data. So they couldn't aggregate the data into a new data lake to then do stuff on top of it, like, machine learning, or just like make it easier to hand people out. Like the US is pretty weird, but like records and all these different things, like whenever I'm having like a medical issue or whatever, and they're like, oh, they're being slow with the records, like I get it. You're probably hitting some like Dr. Seussian thing, and like you're jiggling a handle and you're hoping it comes out, like, because like it's a labyrinth, like you don't really know what's going on, like, and systems are either legally can't talk to each other, or like through bureaucratic whatever can't talk to each other. So it's a Wild West. The for your supercomputer, so like if someone wanted to build, let's just say like I'm learning machine learning. Actually, I have a textbook on that side. I'm learning this because it's actually really exciting, and there's some tools I want to build to save my time. But if someone wanted to build themselves a supercomputer like you did, is this like prohibitively expensive, or does like a university need to like come and be like take care of it?
So let me put this. In Switzerland, things are expensive, so I spent 25,000 on it, just to build it. So it is a lot of money. I think if you understand, and you know, I mean, my postdoc, he was really good putting these all things together, right? It could have been much more expensive than that. Yeah. And also ETH, you know, you have great engineers, so you have help on that side, because you, in theory, it should work, but it doesn't work, and there's a lot of fiddling around till it works. Could you do it for less? Maybe.
I think 25 is pretty reasonable. That's pretty reasonable for a supercomputer, yeah.
Okay, and we also got a grant from NVIDIA, right? So they also supported us with, you know, some graphical. I mean, the hardest is to get those cards, right? Because, you know, they're not available.
They're gold.
That was
they're gold now.
There's this long waiting time. That was I think the hardest part.
Yeah, I feel like it's like I almost wish like companies are to some extent dictating like priorities. You know, if a company was to say like, all right, research institutions like yourself get first go of these things, so then we can have better medicine and all these other things coming out. That'd be pretty cool. But you know, it's more likely like first come first serve. But I do like the idea that, you know, but at the same time, it's kind of like, once again, a company's dictating what you could build, and then it dictates what other people can build. If there is like some type of like weighted measure in terms of like who gets these parts, but because they are scarce, there's like an element of like there's a value hierarchy of like who can get these things. But so it's fantastic that you got one, and I would love to play with it sometime. But I know I'm not allowed to. So that's really cool. So for jumping, you know, like slowly moving back to C. elegans, can you tell us like what a C. elegans is, and then like how translatable, either historically or that you've seen, to its systems to human systems?
Yeah, so I start now with humans, right? I mean, you know, the end goal is to you know, to inform human biology, and nowadays we have better tools, right? We can measure the gene expression, we can measure the protein, we can measure your metabolites, we can measure a lot of things, right? And you know, the thing is, of course, you cannot do experiments with humans. Right, from the bad thing from the scientific perspective. Also, aging takes just too long, right? If I would wait 80 years and see the outcome of an experiment, well, you know, I could do one maybe, right, maximum. And now I'm already too old to do this, right? So then you need. So what we do, we take all these omics systems called systems biology, right? We take the information from there. We come up with different correlations, and these correlations lead to hypotheses, right? And as you know, correlation is not causation. So it looks really good. It's tight correlation, but you have to prove somehow, right? And so that's the reason why we use the elegans, because the elegans is a beautiful system. So it's 1 millimeter in size. These things are hermaphrodites, so they can fertilize themselves. So they are like twins. So it's like having a human twin study, right? So they're genetically identical, the same genes, which makes it a lot easier. And then they eat the same food. They live in the same environment, right? So you can control that everybody eats the same thing. Like everybody's a vegan, or everybody eats only meat, or everybody, you see what I mean? So that's controlled. So you have the genetics controlled, you have the diet controlled, you have the environment controlled, right? And then the good thing is they live around, you know, 3 weeks. And so you can do an experiment within a month. Now, you can also double the lifespan of these animals, and then they live, you know, 2 months, but that's still in a time frame, right? And so we love to use that system as a pioneering system, like a litmus test, to first see, like, you know, whatever gene mutation is associated with longer lifespan, or which drug is there, because then, and then we like to do this in an automated system where basically we have scanners, and maybe I can show you this, because I just let me show you how they look like, the scanners, so you can see this in a sense.
The matrix is back.
The matrix is back. Let me see. So this is you know, in that paper, I think we had a video in there. So here, the picture here. All right.
Oh, cool.
So So what you hear, so these are these little C. elegans, right? And so they are on these little plates, and you put those plates in these scanners here, right? And those scanners take a video every hour, right? And so we have two machines there. We have 16 scanners, so we can run a lifespan of like 10,000 nanomoles at the same time, right? And then we have different interventions. You can do everything. You just have to set it up, put it in there. The machine takes a picture every hour, and then after 2 months or 3 months, you get the data. There's a lot of data crunching because you have to analyze all those pictures, right? But technically, then you get these lifespan curves that look something like that, right? So there's wild type, and then these are longevity interventions, and then you can do all these cool statistical analysis and do those interventions. So this should. Yeah, I think that's what I wanted to show. And
yeah, I mean, if you talk about privacy concerns, these people have no privacy. These little C. elegans, they're watched their entire life.
And so the other thing, the other thing I have to mention is that no, this is an important point, right? Because the next step we go then whatever we find, we go to mice, right? And then in mice, the lifespan takes 3 years, right? So it's a hard time to find a PhD student because PhD student wants to finish around 3.5 to 4 years, right? So we have a shorter point, but in mice, you need a lot of ethical permissions, right? And so again, C. elegans is great because it's not considered an animal, so you could do whatever you want to it. So you need no ethics permission. You can do high throughput screening. Whatever you find there, you narrow down and move it forward. The other cool thing is that you can put them into microfluidics. I'm not sure whether that video is going to be any informative, but if you see these little things in there, these are C. elegans, and we can turn on acoustic waves, and they just trapped in there, and we can measure then how strong they are, right? So you can do a muscle force thing. So you can use microfluidics, so like these little channels. So it's just you can really automate and have really cool things, and you can measure, you can quantify their health span, how strong they are, and all those things you want to do, right? How they move, and also when they're old. For example, here, oh no, this goes too long.
For people listening, we're seeing a video of some wiggly worms. I forgot to describe what was happening at the same time for people just listening in. We're seeing wiggly worms. We're seeing the system. It looked like boxes, petri dishes, etc. And we're seeing graphs of a paper. And if I remember, I'll link it in the show notes.
Good. Yeah, and then here you can, if I get the video to work. So basically,
it's wiggling. It's wiggling on the top right. There you go.
Now there's acoustic wave, and you see this one is an old worm, and you see all these wrinkles, right? It's just
it's gnarly.
It's wrinkly, right? And then on the other side, this is a longevity interventions, and so it's day 29, so they like very close to death already. The wild type worms they're shown here, but then the longevity intervention, they're just still strong. They try to move against the force field.
Yeah, so it looks like a baby one, like a young one.
Yeah, it's two standing waves, right? You can combine physics and biology, which I love to do. And the other ones, now the force field is off. It starts to recover, right? It's a little bit wrinkly. It's really old. It just doesn't move so much. Same age, just a genetic mutation makes it youthful again, right? And so that's the power there, right? You can really then start to quantify all these things.
And so both of those are at late stages of their lives, because you said they last about 4 weeks and they're at day 29. And the one on the right, for people who are just listening, it looked like just like it could be a teenager. It was, hopping around, had a hot rod driving around or whatever. And the old one was just like, I hate being alive, and had like a cane. It was moving so slow.
Exactly. And so then we use whatever we predict in the machines, or from whatever you do, right? We can then try to really test in that system as a first path. And also look at how to stay young, and then move forward in mice. And the next step in mice is then actually take old mice, so 20 months old mice. And then for 6 months, we assess how good they are, like a doctor would do, like, what's the grip strength? How does the hair look? Is it gray? Does it have kyphosis? How does it move? And other stuff. So there are like preclinical parameters are called. It's called the frailty. Like, old people are really frail, right? You can measure that also in mice. And then look where the interventions we found in the computer, then in C. elegans validated, and then move it forward, whether that works in mouse, and then you have a pretty good chance that hopefully it also works in humans, right? So that's then the next step.
It's fascinating. I've been wondering if there's a way to like artificially make rats, have a shorter lifespan, so you could do faster research on them without giving them a disease. So then you could have the same type of rapid testing, and then make it a little happier for PhD students, I guess. But
yeah, so their disease is called progeria, so they live shorter, right? It's not the natural aging process itself. There are other people that try to make systems like this in mice. Again, it doesn't confound. It's not the natural aging process itself, and so somehow you need to find. I mean, what is cool and new, and one way to overcome this are these killifish. So these are fish that live for 3-4 months because they're in Africa. They live in these ponds that dry out, right? So for a short time, and so those ones are vertebrates, right, and more closely to it, and that's the new system that's actually set up now to look at these things, that to have a closer, so the gap of translation is not so high from whatever model organism go to humans, right? And so people trying to come up with new systems, so to speak. Again, biology is so complex, you need to find something. I mean, the dream would be to do this all in organoids, right? Having human organoids and have a way to model aging. But again, it's in cell culture, aging it's just different, right? And then it's into organ specific. So you would have organs on chip, all different organs of a human on a chip to do this. You have the brain organoids, you have intestinal organoids, you have whatever. Then you put some immune cells in there. I mean, people are trying to work on this, and if that hits off, that would be fantastic to study actually human biology.
Yeah, I've seen stuff coming out of UW. I forget which one. There's one in Chicago, and then there's the VIS in Boston. They came out with some chips, but it's usually like a localized chip, a biology chip, where it's like they're replicating the uterus or they're replicating a kidney function. They're not they're not systems integrated together, which is well, maybe like one day they could build up to it. So it's good that they can do one thing, but then how do those systems interact with other systems also has like a play into it as well.
Exactly. There's a lot of things going on, but I mean, big pharma companies already recognize this. For example, Roche in Basel, Switzerland, they built this Institute of Human Biology, and they're using human organoids to do this, right? So Roche recruited Hans Clevers, right, one of the forefathers, right, of organoids, and also then Matthias Lütolf, right? And so that's fantastic to see that their movement in that science, right, to do this exploratory research in there to get it to the next level. So I think there's one of the future things lying there. It's a lot of work ahead, right? And to me, unfortunately, the external matrix is very hard to model in the organoids. So I watch it closely, right? I would love to jump into it. It's just not there. It's just too far away. For me, but it will.
Is there a component or aspect of the extracellular matrix that is difficult for these organs on a chip to track or to replicate?
Yeah, because it's so you have different cell types in a tissue, and every cell type makes a different part of the matrix, and it's secreted, it comes together. It's just it's a very dynamic system, and that hasn't been really like figured out in a detail that reflects in vivo system itself, and that's the problem. I mean, the amazing thing is that you can take say skin cells, and put it into wells, right? And their job is actually to secrete collagens, and they will do this all the time. They secrete collagens, but since there's so much liquid, it just goes everywhere. If you crowd the system with big sugars, right, and the collagens stay closer to the cell surface, then the external matrix starts to build. So they start building, the collagens will start to integrate. This builds the matrix, it grows, right? And then there's a remodeling process happening within like 6 days. It starts remodeling and starting to adapt. So a fat cell makes a different extracellular matrix than a skin cell, right? And it's just it's amazing to see. And to me, that was actually, I've been trying to play around with then again, but again, the modeling part of aging is the problem. But what I took away from there is actually that there was a saying in the extracellular matrix field that, every cell makes their own extracellular matrix, right? And I was like, okay, but, nobody has really shown this, right? And nowadays with the single cell sequencing, right? I was thinking if that's really true, then you could take single cell sequencing data, right? And predict cell type just based on the extracellular matrix. And so I got together with an old friend of mine, Patrick Chop at the University of Basel, and we looked at limb development, so in mouse and in chick. And he had the single sequencing data and doing development. And what was clear is that when the cells are less developed, less differentiated, the extracellular matrix, and we're talking about around 150 genes, right? We just use, right? It's not so good predictive factor, but the more it gets, the cells that get differentiated, the better predictive factor has the extracellular matrix. So from the whole transcriptome, from single cell sequencing, the extracellular matrix around maybe 2% of the transcriptome. And that 2% is sufficient to say which cell type it is. So we can predict just on the whatever extracellular matrix gene is expressed, we can say, well, this is, this is going to be fat cell, this is going to be a muscle cell, this is going to be this cell, right? Which is amazing. So basically the extracellular matrix mirrors the cell identity. So you can use this as a readout. What is also was interesting to see is actually that it mirrors developmental stages in the cell. So we can say what stage it is. And I took this a little bit further and thinking about, there were people like Alexandra Nappa that were saying that, you can predict cancer type just based on the extracellular matrix composition, right? And so in the end, I put this all together and I called this the mature type. So really the composition of the extracellular matrix can, that is caused or associated by a phenotype or by cell identity or by, health of the disease, right? Or longevity. And we use this actually for drug prediction. And so we, maybe I show this real quick if that's one thing we learned. Let me show this real quick, because that came out of these very theoretical biology thinking, right? To just look at things and combine it, right? And so, what we've seen is that we can actually. So what we've done, we've took the whole expression of different tissues in humans, right? And just said what is an old extracellular matrix expression and what is a young one, right? And so we have a signature how a young extracellular matrix should look like, right, in any tissues. And then we overlay this with drugs, a gene expression. So you can give a drug to a cell and it gives you a gene expression, right? And we ask which drugs have a young extracellular matrix gene expression. And we identified 180. And then we went on and used the Elegans to actually do the lifespan, right? We predicted different drugs and see, whether they could actually extend the lifespan. That's shown here, for example, in black. That's the control. We can feed chrysin as a part, or dapsone, or vitamin E, vitamin B12, and it increases lifespan. So basically, we were able to repurpose in drugs that are out there for other things or supplements to really predict these have longevity benefits, right? And that was, that's one way in there, and it really is. It's amazing to see that there is, such a predictive power in there, not only for the cell type or disease type a cell is, but also whether it's reprogrammed for longevity, right?
Yeah. I'm wondering what would this type of process look like in correlation with Michael, like he's getting like his ears must be ringing, but like Michael Evans' work where with the bioelectricity, or if you combine those your two approaches while looking at like an axolotl limb regeneration. I'm very, very, I'd be very interested to see like what would come of that. I mean, the correlation, not causation and stuff, but I'm just curious, like what hypothesis we could generate from what that would look like. What is the future of your work? Like when you do this research, is it your contention to build out theses that people could translate into drugs? Is it the purpose to build out understanding that people can then build off of to make drugs and interventions that help people, and be like the idea maker? Or how do you see yourself and your research moving forward? Like, what's your ambition for yourself? I guess.
So I have two. Yeah, no. I have two things that are close to my heart. One is to I went into biomedicine because I'm interested to bring something good to the people, right? That's where I started actually my journey in my studies, right? So that was always my goal to apply it in a sense. On the other sense, I love to think about things, to do the theory aspects, right? And at the moment, the exo matrix part is really geared to drug discovery in a sense, right? So that is the end goal. Like, I was looking for a mechanism that is understudied in the aging and longevity field, right? And then really try to figure out the mechanism. And I think now after 7 years, we have a really cool observation that we made. So we found actually that those changes in exomatrix are read out by these physical change in stiffness, right? A forced transduction and changes gene expression in nucleus. It's called mechanotransduction in a sense, right? So we've seen that these longevity interventions really, require mechanotransduction. It's like an override signal. So you don't get any longevity benefit, right? So that, and we try to dissect, very explorative, what are the different players there in the pathway? And we map this out. So we really have mechanotransduction that's signaling downstream to the SIAP transcriptional co-activator, then, helps in reprogramming of the cell, right? And so that would be one way of a drug target there to move this forward. And so I think that is just an understudied field with the great potential, right? And so we really try to find drugs in a sense to move it forward, right? The second part is just like, I like to debunk different things. And so there's everybody's in the field there and say like, we want to find these interventions, but when do you want these interventions to work? I mean, to me, I would love to be like 75, right? And then start to use this intervention. But there's no evidence that it ever worked in an old organism, right? So because most studies are done actually in young organism or middle-aged organism, right? But nobody has done these things when the organisms are super old. And so I'm just going to show this one because I think it's easier to understand. This time I try to make a better job explaining. Let me see if I can show things and else explain it. And so that's another paper. And so to get at this question, so there were technical challenges. That's what in the beginning we were saying with the method. So we had to develop a new method. And the method here, so this is a little bit engineering, so please stay with me there, was a system. It's called the Oxen degradation system. That's a system that's used in plants. And we tackled one important gene. That's the insulin IGF-1 receptor that has been shown if you have a mutation in that particular gene, you can double the lifespan of the Elegans, right? And so, because if it's a mutation, the problem is that it's always there, right? It's not, you cannot just switch it on at the old age, right? There's another way to do it. It's called RNA interference. You knock it down, but that only works till day 6 of adulthood. So very only early adulthood. Say when you're from a teenager till you mid-20 or 30 or something like that, right? But we wanted something that's super, super old. So we're thinking about this system, this Oxen degradation system, and basically how it's working is that you tag your insulin IGF-1 receptor with this sequence called Degron. And if you have in the cell another protein called TO1, it will go to the stegron only if Oxen is a plant hormone when it's there and starts to polyubiquinate it. So it tags it. It tags that protein for degradation, and then the system will come and then degrade the whole protein. And so that works very well inside of the cells. So within 30 minutes, the protein is just gone. So basically, so think about the cell, right? You can actually just give that hormone to the cell, this plant hormone, and it will just degrade your protein within in 30 minutes. And so that was the basic idea. And honestly, I first thought it will never work because it's a transmembrane protein. But you should never talk yourself out of an experiment. And also the student was very persistent, and I just didn't want to talk about it. But it worked. So we were able to do this. So we were able to degrade this. And so what we can do this now, right? We can degrade it. And I want to show you, that's the key experiments I want to show you here. So what you see here, this is the lifespan plot. On the y-axis, you have the population life, 100%. And on the x-axis are the days. So here, 20, 40, 60 days. And so in plaque is just, the wild type control. So you see they live around a little bit more than 20 days, 30 days, everything is done. It's good. And then you can actually then use the Degradation system and degrade the insulin receptor. And you see this is the longest curve. So they live almost past the day 60. So we're able to double the lifespan. So this is all nice. It works right when they're young. Now we ask, could we push out the system here? And so here, I don't know if I can make this bigger.
No, but I actually went bigger. I'm like,
yeah,
earlier.
This time works. Different journal, right? And oh no, this is the wrong picture. We looked at which you can also do it tissue specific. Sorry, too excited, too fast. Wrong figure. So we can also figure out what tissue it is. That's not the figure I wanted. So this one I wanted. All right. Now, again, yes, same game plan, but now we're gonna start when they're very young with Oxen treatment, right? And then with the days. So in dark blue, so the control animals, when they don't get the Oxen treatment or degradation of the insulin receptor, they live again for 25 days or mean lifespan, and then by 30 days everything is dead. We do it when they're very young. You get a maximum extension. Again, you'll go up to almost day 60 again, right? You get this doubling, right? And then you can do at day 7, which works very nicely. And then it gets even more and more. And so even day 14, you start day 16 and day 20, right? And you see, the population is dead. They're all dying, right? And you give it. And here, this is what I want to show you. And that's the part, this was the killer experiment, right? And so what you see here, so these are the animals. And at day 21 of adulthood, right? What you can see, 75% of the population is dead. So they all died, right? So only 25% surviving. The worms that don't get the Oxen to degrade insulin receptor, they die within 4 days. So day 25, from day 21 to day 25, all of them are dead. The last 25% died. The worms we're gonna give the Oxen to, right? What you can see is actually this increase. And so it goes past day 30, it goes past day 35, and there's one that's 20, it's, almost another. So if they start 21, it's almost hitting the 40 mark. So that worm was able to almost double the lifespan, which is, if you think about it, remarkably, because like some of them actually were able, close to death, really close to death. We do this intervention, we degrade the insulin receptors, we were able to double the lifespan. And,
it's weird.
And it's so cool. And we're trying to now figure out, like, how is that possible, right? Is what's happening here? Is it actually that, aging is stopped or extremely slowed, right? So in a sense, right? And they just age slower. Are they rejuvenated? You could think it's just a one-time treatment, right? You could think like, okay, Zach is like the Yamanaka factors. They rejuvenated, and then they live for another, 21 days, right? Because that's the normal lifespan. It said it's like they have the whole life again. And so we're trying to figure this out, and we do this with a collaboration with a lot of different labs. So this finding has been reproduced by many different labs, and we're trying to debunk what is happening, right? What is repaired? I can tell you already that some proteins that aggregate with age, they actually, these aggregates are remodeled out. So get loose, you get rid of them, right? But not all the things work. So, sometimes you improve swimming, how good to swim. Other times, it's not working. So differential pumping, how much they eat, it's not going to rejuvenate. So there's certain parts that are improved to rejuvenate. Some parts are just slowed down or not get worse. And so we try to dissect this, what is actually happening in vivo in very, very old animals. And I think this is really exciting because that gives you some insight what we could do, right? I mean, what I have to say, what's not better is they still look old and ugly. They're better, they're more stress resistant, they can move better, right? They're more fitter. So you could imagine that you're 75, you get this intervention, you still look old and ugly, but your muscle performance much better. It's like say like a 50-year-old or a 40-year-old, right? And you're more resistant to stress. But unfortunately, you not get beautiful again. Let's put it this way.
Yeah.
At least if you're worm, right?
If you're worm.
So
yeah.
I talk about humans. Let's say this is all in worms, right? Okay.
Yeah. You do in rats, and they grow out like really long blonde hair. Start like looking like supermodels or whatever in the rat world. But that is really weird to look at the graph and see that at that intervention that it almost had the same effect if you did it earlier, because I would think there'd be some type of buildup or some type of like senescent buildup that would like, okay, you have an intervention, like there's an uptick, but then I would just, it would go right back down like a meteor shower, like just a meteor going right back down. But it almost like plateaus correlatively to like what happened in the early stages. That's just really weird. Like I almost makes me think that there's some level of rejuvenation happening, or some level like senescent I don't know, getting rid of. That is really weird. That it looks odd. I'm very curious to see what comes from your work on that, because I would assume like maybe like it added like 12% or 15% onto it, which is significant when you're at that time. But it's like when someone's falling, if you try to help someone, they're like an inch off the ground, I wouldn't expect you to be able to pick them up and like get them to move 10 feet when you're just trying to get them to stop from dying. Like it's weird that not only did you get them to stop from dying, but you actually moved them. You gave them like locomotion to move them into the future, like almost doubling them. That's really interesting.
I'm so happy you think this way. You got it right. That's the mystery we're trying to debunk, and it's extremely exciting. And the more people I talk to, right, something they're just resistant to dying, right? Because dying is actually also a biological process, right? There's so many things now, ideas you could test, right? Again, this is very on the basic science part, on the conceptual part, because you could learn so much about the aging process itself. And it comes back to the thing you said, with the methods, right? We developed a method there, or adjusted a method there, and built a system. We could address now these fundamental questions. And I'm really happy to hear, from a non-scientist, right? Not thinking about worms to get excited about, because, one of the biggest challenges I face, for example, is, what is the relevance to human biology, right? And I have to tell them, this is more the theoretical part, right? Think about on theoretical terms that what is the aging process and how can we, reverse, or what can we intervene? What's happening when we intervene, right? It just, there's so much biology that can be detected that we could exploit for later. And who knows? I mean, it might be conserved in humans, it might not, but you learn a lot about biology. And I think, to me, as a scientist, it's important to produce knowledge. In German, the word scientist is called Wissenschaftler, somebody who produces knowledge, right? And so this is more on the producing knowledge kind of thing, right? Once you have the knowledge, I mean, even Musk builds these rockets and they failed, and he says, well, you learned something, right? This building knowledge, right? It's not, and then you do next iteration and next iteration. I think these are these kind of things that are needed for translational medicine, in a sense. So I'm really happy you quickly understood and saw exactly the excitement there. And so if anybody wants to work on it, we deposit those worms in the system. It's called the CGC, where they have a repertoire of worms. So any researcher in the world can go and ask for those worms, and they will ship to them. I think it costs them $10. It's just a shipping fee from the thing, but it's all we all give it back to Sense, and everybody can try and try different things and work with it, and shoot me an email if you have questions or anything like that. But it's also all described in the paper. So I'm just trying to get more people excited to, because I think it's an important problem to figure out.
Yeah, I think it leads to like an Einsteinian like universal theory of biology. You know, at the end of Einstein's life, he was trying to unify all of physics, and it feels like with what you're doing, that it might put another piece of the puzzle in that gives us a better picture of what's going on, and then you can translate that in many of other different ways. Yeah, it's really compelling. I'm very interested to see what comes from it. Does the I forget the technical term. I think it started with the D, but does it work with all proteins, or was it just the proteins that, or specific proteins, the intervention that you described a minute ago?
So here we tackle just one protein, and it's called DAF2 in C. elegans, but it's the insulin HF1 receptor in us. So it's the precursor receptor, right? Evolutionary same, but it's distant. It's much simpler. C. elegans is much simpler. So that's also the reason why we use this. So it just combines this one. And it was actually one of the first one to be discovered. So Cynthia Kenyon had done this work in 1993, where she's shown that those worms live twice as long compared to others, and they're youthful, right? Also, the worms you saw there in the video, they're moving, right? These are also these DAF2 mutants. So it's just the right target. It's for C. elegans. It's the right target for C. elegans. It's going to be different for human biology, right? But it's just the same. The same concepts or mechanisms are reused in evolution over and over again. They built this networks and the circuits, and they're just adapted in different contexts and reused, right? So if you figure out what to look at and how the interconnections work, then you have an easier time to figure out this in a new system.
Yeah, I think the crab as a form has been re-evolved in different areas without any association with there's not like one main crab line. Like if we see aliens, they might all look like crabs, because on Earth, like the crab structure of like the legs and the pincers and stuff keeps getting remade in different areas without any relation to each other. It's just like, oh, this is the great adaptation for that area, and it just happens to be this form, which is kind of weird. And
it works, right? And one thing I want to mention, now you mentioned the crabs, is just aging itself. I mean, there's no biological reason why we should age, right? And it just happened during evolution that one of our first ancestors was aging. So if you think about single cell organism like bacteria or yeast, right? Single cell, but they also age, right? I mean, you think about bacteria or yeast, what they do, they grow and they bud off, and then they grow and they divide and grow and divide and bud off, right? And so during that time, the mother cell, so to speak, with time we accumulate damage, molecular damage. So the mother cell retains that damage, and then the daughter cell, is all more youthful, right? And that's why, because the mother cell accumulates the damage, it starts to age, right? And somehow it just happens that aging was there, and it's across. It's so much conserved that you can use these simple systems to find some truth about aging, or some concept or theories or hypotheses about aging, and then it's mostly applied then to other systems.
Yeah, I was wondering if the intervention that we talked about, if they could have any effect on prion disease, where like prions like misfold and like go crazy and like cause problems, which there's no real treatment for that. And it bothers me as someone who has to take a blood product, getting the brain eating disease. But I know we're short on time, so I'm trying to consolidate a bunch of questions into two, so I can get you out of here without eating up your day. So what books would you recommend people check out? I'm as you can tell, I'm trying to look for more things to read. I'll add it to the newsletter too.
Oh, I have I have a passion for books, especially for science and in the aging longevity. At home, I have a huge bookshelf. I don't know. That's a hard one. I mean, that's a hard one. From the top of my head, I would.
It doesn't have to be aging related. Just for the record,
okay.
It can be anything.
Okay, so if you're into systems biology, theories of aging, Uri Olan has a really good textbook kind of thing for systems biology. That it's very technical, right, in a sense, but to me, it was fun to read. And then, I mean, there are lots of books about aging out there. Most of them are really good and a fun read. I also like, I don't know, from 0 to 1, like stuffing in the startup world, right? Things about problems, so like that kind of book. I don't know which. Yeah, it's too. There are too many. I cannot. I mean, also, I mean, George Stewart's Regeneration book, right? I mean, you had him on the podcast. I mean, definitely that is, a classic everybody should read, and it's eye-opening. But what's all out there? I think these books, I would say.
Sweet. And then I'm gonna ask one fan question, which there was a bunch that people wrote in, but unfortunately we got distracted with machine learning, and it was exciting, which is all good because like we can go down any avenue, but also don't want you yelling at me for going over. So the cell improvement matrix. Could epigenetic? This is from people have crazy names. Grizzletown. We're asking your question. Could epigenetic reprogramming of cells somehow improve the extracellular matrix during aging?
Yes. I think so, because it's linked again. If you think of the exo matrix, right? So there are this network, and then it's held up by receptors, right? And it's pulled, and so there is the acting cytoskeleton going to the nucleus. And so basically, if you pull on the matrix, you pull on the nucleus, right? Which changes the nuclear envelope and changes the epigenetic landscape. So if that goes wrong with aging the epigenetic landscape, right? If you improve that one, it will restore cellular function, and then it will restore the extra matrix again. And then, so you can go both ways to start tackling there. So yes, and it's interconnected. It's really interesting if you look at electron microscopy photographs, right? Of cells and tissues, you cannot tell which one is the out and the inside because it's so crowded, and you see all these bundles. And only if you see the cellular, the organelles, you say that's the inside, the outside. It's just amazing to see how they interconnected. And I just want to point out again, the physical forces have a signaling component in there.
And then in the next 5 to 10 years, what advances would you expect to have for your life? For longevity or health span that you're watching, what do you think people should reasonably expect?
So what I hope is going to happen are clinical trials for aging, right? For healthy aging. And so the reason why I say this, I mean, we have now supplements, we have some drug candidates, some interactions. Particularly, they work really nicely. We know they rejuvenate mice and worms and all those stuff. But we have no idea whether they work in humans. Even dietary restriction is still unclear, right? So what I would love to see that we have clinical trials to show first that it's safe, right, in humans, and effective. I want to see the efficacy there, right? And that would be wonderful. And I think lots of people are working on there. It's a lot of work. And also one part of my effort is geared towards that. It's not so easy because aging is not a disease and what you read out, but it's an exciting field to move forward. And I think that's really important in the next 10 years that we have something like that. And then you can push through those interventions we know that work in preclinical models and mice and everything. We can also translate to humans and make sure that we don't do any damage or any harm, that they're safe, less or minimum side effects, so you can calculate, but we know they're effective, so that they work.
And then for your work, as the last question, what do you need help with? Is there anything that listeners and myself or anyone can help you with your work?
Oh, there's always, I mean, I get, lots of creative people actually contact me and we start discussing things. So I have people actually, coming from programming side, or they're working on airplanes, for example, right? And we're discussing a problem, and they start moving projects further, right? So nowadays, even if you have a passion for longevity and for science, you don't need to be in a lab itself. So whatever you know best, right, can be used, right? In a sense, there are so many problems that need to be tackled. It doesn't even be research. It could be somebody who is interested in longevity and is a lawyer, right? And then how there are so many questions I have, for example, if I have something, how do I spin this out of a lab? How do I do these deals? How do I connect? Because, for example, what's happening, whatever happened in my lab, right? This is on the academic side, where the innovation is happening. But in order to move this forward, it has to be spin out of the lab into a startup, right? In the startup, it has to be further developed, de-risked, and all these furthers. So then that, bigger companies, pharmaceutical companies get interested in, because they see, okay, there's the first proof in humans, right? And then they take this over to move it really forward, the translational pipeline, because it's a long process and it's a big process, right? So we need the interaction needs. So those steps need. And so all of those steps, if you see you have a capability to help, then yes, please. That's not only my lab, there are other labs also. Just start the conversation. I think most scientists nowadays, they're really open to talking to different things and having people involved, because we have so many problems. Because you're trying to start to build the whole pipeline, right? You start from the beginning. There's so many things you cannot solve, and if you have somebody who has the expertise and wants to help, it would be fantastic. It takes a village, right, in the end.
Sweet. And then links to Collin's website, the places to read up and potentially reach out, will be in the show notes. Collin, I want to thank you for coming on the show. This has been Learning with Lowell. We've learned a lot today. Everyone who's listening, say something in the comments. Let us know what you got from this, and if you see the power of what we nerded out about 7 minutes ago in terms of the intervention late in life, and yet still having the effect of massive extension, which is weird. Anyone have any hypotheses about why that is? Put that in there. Then hey, maybe we can test them out. We'll have a bit on this or something. But Collin, thanks for coming on the show.
Thank you so much for having me.